On behalf of our International Pediatric Hodgkin Lymphoma Collaborative Study Committee, representing the Children's Oncology Group and the Urinet PHL, I am pleased to present subgroup analysis focusing on the response and toxicity rates observed in younger patients and primary refractory patients with relapsed Hodgkin lymphoma enrolled on the CheckMate 744 trial. My disclosures are noted here. My institution receives research funding from Merck through a subcontract with the Children's Oncology Group. Over 90% of pediatric and young adult patients with Hodgkin lymphoma are cured. Our challenge, though, is that among the patients that either don't respond or relapse following initial therapy, relapse or progression following autologous transplantation for retrieval therapy is still a major concern, with up to 45% of patients experiencing subsequent events at 10 years, which is demonstrated in a CIBMTR analysis, which is demonstrated on the left of this slide. While long-term outcomes are improved in those patients that achieve a complete metabolic response prior to autologous transplantation, the approaches that are used to obtain that remission are often associated with increased treatment burden in this younger patient population. And as shown here on the right-hand side of the slide, that over half of these patients are dying from complications other than relapsed Hodgkin lymphoma, such as second malignancies or cardiac failure as a complication of the treatment they received. Combinations of brentuximab vedotin with nivolumab or bendamustine have been associated with high overall response rates and complete response rates in phase 2 trials in adults with relapsed and refractory Hodgkin lymphoma. These regimens are expected to have less long-term toxicity compared to conventional chemotherapy regimens such as ICE, and have the additional advantage of being suitable for outpatient administration. CheckMate 744 is an international phase 2 study for pediatric and young adult patients ages 5 to 30 years with first relapse or refractory classical Hodgkin lymphoma. The pediatric group was defined as less than 18 years of age. No prior checkpoint inhibitor, bendamustine, or autologous transplant was allowed, although prior treatment with brentuximab was permitted. Risk stratification consists of 2 cohorts, R1 or low risk and R2 or standard risk. This presentation focuses on subgroup analysis of the R2 cohort, which met its accrual goals in May 2018. Accrual to the R1 cohort is ongoing. Risk category is determined by stage of diagnosis, time to relapse, and clinical features at the time of relapse. Patients with advanced stage of diagnosis, early relapse, B symptoms, or extranodal disease at relapse, or relapse within a prior radiated field were enrolled in the R2 cohort. The study design is illustrated here. Patients received 4 cycles of nivolumab with brentuximab. After 4 cycles, patients who achieved a complete metabolic response based on blinded independent central review went on to high-dose chemotherapy and autologous stem cell transplantation. For patients with less than a complete metabolic response, 2 to 4 cycles of brentuximab with bendamustine were administered. Consolidation therapy was then administered based on response. The primary outcome of the study was complete metabolic response, defined as Deauville score less than or equal to 3 by blinded independent central review of FDG PET scans at any time point prior to transplantation. The data that is being presented today includes the pediatric cohort and the patients with primary refractory disease. The baseline characteristics are presented in this table. 31 or 70% were less than 18 years of age, and 24 or 55% were primary refractory, of whom 17 were also in the pediatric cohort. High complete metabolic response and overall response rates were achieved in both subgroups as demonstrated in this figure. 58% of the pediatric group were able to proceed to autologous transplant without additional alkylator exposure during the reinduction therapy. And then 87% achieved a complete metabolic response prior to auto transplant with this treatment approach consisting of induction and intensification. Similar results were shown for the primary refractory group. 63% were a complete metabolic response after the 4 cycles of nivolumab with brentuximab, and 83% achieved a complete metabolic response with the additional cycles of brentuximab with bendamustine. And these results were very comparable to the cohort as a whole. The regimen was well tolerated by the pediatric patients with relatively low rates of grade 3 or 4 treatment-related adverse events during the induction, as demonstrated in this table. Only 6 patients experienced a grade 3 or 4 treatment-related adverse event, all of which resolved. Only 1, treatment-related neutropenia, was a grade 3 serious adverse event. Unlike experience with conventional induction regimens, no treatment-related febrile neutropenia, thrombocytopenia, or anemia was reported during induction. And importantly, no pediatric patients had discontinuation of their therapy due to an adverse event. Similarly, immune-mediated adverse events were infrequent and of low grade in the overall cohort and in the pediatric cohort too. As shown in this table, no grade 3 or 4 immune-mediated adverse events were observed. Infusion-related reactions were also infrequent. In the overall R2 cohort of 44 patients, a total of 10 or 23% of patients experienced an infusion-related reaction, including 2 grade 3 or 4 events. The same percentage of pediatric patients experienced infusion-related reactions, although all of those were grade 1 and 2. In conclusion, high complete metabolic response rates were observed in both pediatric patients and in primary refractory patients, with rates comparable to the cohort as a whole. The combination of nivolumab with brentuximab vedotin was well tolerated in pediatric patients with low rates of immune-mediated adverse events and hematologic toxicity. The data from this study supports consideration of this regimen in young patients as well as those with difficult-to-treat primary refractory disease. And the regimen has the added advantage of outpatient administration. Enrollment to the low-risk cohort is ongoing. And finally, I'd like to acknowledge the patients and families who made this trial possible, the clinical study teams who participated in the trial. Study drugs were provided by Bristol Myers Squibb in conjunction with Seattle Genetics. The study was supported by Bristol Myers Squibb in conjunction with the Children's Oncology Group and the Urinet PHL. And thank you for your attention.